restriction compelled us to celebrate this auspicious day online. This year, 2022, we could assemble in Acharya Bhavan. Okay, our Acharya Bhavan and celebrate it offline. At first, we pay our heartfelt homage to Acharya Bose, a doyen of Indian science who made during the colonial period epoch-making contributions on microwave, produced 5mm wave in 1895. This is the basis of all modern wireless communication, including 5G. Similarity between living and non-living, cosmic unity, plant research, monumental studies on plant electrogeology and neurobiology. Taking cue from his plant research, investigation on consciousness has been started recently. Today's speaker is Professor Majumdar. We are grateful to Professor Majumdar for agreeing to deliver 164th birth anniversary lecture of Acharya Jagadis Chandra Bose today. Title of his lecture, I quote, and comma, as we moved, comma, we embraced and absorbed, unquote. A narrative and contextualization of the work of Savant Prabhu, this year's Nobel Prize awardee in physiology and medicine. What an astounding title. We all look forward to hearing it. Professor Majumdar, perhaps most of you know, but for our new friends, Professor Majumdar, PhD, FNA, FNS, CFT, WSA, National Science Chair, Distinguished Professor and Founder of National Institute of Biomedical Genomics, Emeritus Professor, Indian Statistical Institute, Emeritus and Adjunct Professor of many Indian institutes in India. He is the President of Indian Academy of Sciences, Bengaluru, 2019 to 2021, visiting professor, genetic consultant in many national and international institutes and organizations. Regarding his award and honor, if I cite all, I cannot finish. I will eat up his lecture time. Many. He is the elected member of the Council, International Human Genome Organization, Hugo, for a couple of times. Elected member, mind that. His contribution made very significant contribution in the field of genomics, genetics, statistics, better known as Gene Guru across the media. One of his major interests is human evolution. He worked extensively to uncover genetic architecture behind disease susceptibility and drug response. He is one of us, a trustee of the Sajjasi Bose Trust. We are working together since 2009. We, the trustees, mention with gratitude the kind patronage of Professor Majumdar and his institute, National Institute of Biomedical Genomics, we receive to save Bose's legacy treasured in Acharya Bhavan, and to save our mission, the Science Heritage Mission, as of this subcontinent. Now, I would like to tell a few words about Acharya Bose, Acharya Bhavan, and Jesse Bose, just about our new friends. Acharya Bose is a scientist par excellence, a quintessential polymath, a literator, a philosopher, a connoisseur of fine arts. I quote, the avant-garde of the subcontinent, first science fiction writer in Bengali. His genius transcended boundaries. Acharya Bhavan. Bose built his historical residence Acharya Bhavan 1902 at 93 Upper Circular Road, now Acharya Chandra Road, where the Bose couple lived till their last days. So it's a 120 years old Grade 1 heritage building of the city. Acharya Bhavan is a treasure trove of many personalia memorabilia of the Bose couple, many artifacts of historical importance. It is a rich repository of Bose's original instruments, mainly plant research, books, manuscript, etc. Many of his path-breaking experiments on responses of plants to various stimuli, radio waves, cosmic unity, etc., were carried out in the laboratory in Acharya. Still, it is here. Acharya played an important role in the nationalist movement during the Bengal Renaissance. Bose had played host to many luminaries of the country, including Gurudev Rabindranath Tagore, Swami Vivekananda, Sister Nivedita, Mahatma Gandhi, Acharya Prafulla Chandra, his notable students, S.N. Bose, Meghnad Saha, P.C. Mahalanobis, S.K. Mitra, D.M. Bose, etc. His interaction with world luminaries like Albert Einstein, Bernard Shaw, Romarella, Lord Raleigh, Henry Cavendish, Okakura, to name just a few, add to the glory of the Acharya Bhavan. Our trust restored the first national flag designed by Sister Nivedita and paintings by the renowned artist Nandalal Bose and the contemporaries. There are many antique furniture, rare sculpture, collection of Bose, and many gifts from world luminaries. Today, Acharya Bhavan stands as a monument to the multifaceted genius of Acharya Bose. It is the life and work of Sir J.C. Bose, vis-a-vis the history of that era, that is treasured in Acharya Bhavan, where science, arts, philosophy, nationalism converge seamlessly. Acharya Bhavan thus has an illustrious past and heritage which should be preserved at all cost for posterity. J.C. Bose Trust, a trust founded by Bose himself, has been entrusted to maintain Acharya Bhavan along with its movable and immovable properties, to preserve Bose's legacy and heritage treasured in Acharya Bhavan. This is separate from Bose Institute he set up in 1917. Mission of the Sir J.C. Bose Trust, mission of the present trustees is to develop Acharya Bhavan into world-class science heritage museum in memory of Acharya Jagadis Chandra Bose, just as Albert Einstein House in Bern, William Shakespeare's house in Stanford-on-Avon, and Marie Curie's house in Warsaw were converted into museums. We appeal to all to support this great heritage mission of the nation. Thank you very much, Professor Mujumdar, and your organization, NIMBIC, our advisory board, my colleagues and staffs, all participants and visitors today for making the program a great success. Today, our trustees extend cordial invitation to you all to visit Acharya Bhavan and support our great mission to save the legacy of Acharya Bhavan in Acharya. Legacy of is a great heritage mission of the country, of the nation. Thank you all. Okay. Now I request Professor Majumdar to start your lecture, please.
Thank you very much, Professor Chakraborty. This is very, very kind, and I didn't deserve all that. Let me share my screen if I can. I think I cannot share my screen until my host enables me to share my screen. What I see is that you have disabled my sharing screen. Who is operating the website? Ask me to give this talk. The title of my talk might sound a little enigmatic. I will clarify as we move along. This is all about human evolution, which is essentially what this year's Nobel laureate, Nobel Prize winner did and got for his contributions. What I was asked to do is explain why this gentleman got the Nobel Prize, but I thought that if I just provide a narrative on what he did without contextualization, it probably would not be understandable to most of you. So what I decided is that I will first provide a contextualization of the work that's been done by Subhandev Ketu, who is this year's Nobel laureate in physiology or medicine. reason why he got the Nobel Prize is that the Nobel citation for his discoveries concerning the genes of extinct hominins and human evolution. So this is kind of a little strange because he has gotten a Nobel Prize in physiology or medicine for his studies and for his discoveries concerning human evolution, which is quite soft and a little bit of a disconnect. But his work is so deep that I think the Nobel Committee was lost where to place him. And I think, as I will share with you the last slide or last couple of slides of my talk, the implications of his discoveries have profound bearing on theology of medicine. So I will give one example of how it has implications on theology of medicine. Be that as it may, the work that's been done by other people is very deep and is worthy of let me contextualize it. Like I said, that it is on human evolution, and I'm going to start about 5 or 10 million years ago and quickly come to recent times, talk about humans.
May I interrupt?
So I will move along. Like I said, and that's when our story begins. And what happened at that time, we were apes in Africa, and then the extinct species. One of these species led to modern, which is the right-hand side of this particular, tree. So that's the root of the tree at the top, and that's when we were all apes. And then one particular branch led to the modern humans. The other branch continues, and there evolved many other kinds of, species such as the gorillas, the chimpanzees, the bonobos, and so on and so forth. So we will concentrate on this right-hand line of this particular tree, which is leading to the modern humans. And the entire story will revolve around this right-hand branch of this tree, again, talking about human evolution. I'm going to jump. I've jumped already for 4 to 6 million years. And at that time, about 4 million years ago, after we evolved and we are now walking down that right-hand path of that particular tree that I showed you, we became bipedal. And we started to spend most of our time on 2 feet. And this had profound implications in terms of our survival possibilities, in terms of our evolution. How do we know that we became bipedal? Well, we know today we are, of course, bipedal. When did we become bipedal? I said that we became bipedal about 4 million years ago. How do we know that? And there are various kinds of evidence that tells us that we became bipedal about 4 million years ago. But one of the best pieces of evidence came from this particular volcanic ash bed, where somebody who walked on this volcanic ash bed, even when the lava was not completely, solidified, that particular animal who walked on this volcanic ash bed was a bipedal animal. Most of us probably won't be able to read the footprints, but those who are experts in reading footprints will vouch that whoever walked on this piece of volcanic ash was actually had a bipedal gait. How do we know that this was about 4 million years ago? Because the volcanic ash bed can be dated using various kinds of radioisotopes, etc., and it's been dated to about 3.7 million years. This volcanic ash bed was discovered in 1975 by a member of a family that spent generations discovering or investigating evolution in Africa, and that's a leaky family. And there are multiple members of the leaky family, each of whom is a very famous evolutionary biologist, a paleontologist, and so on. So they have actually discovered this. Mary Leakey discovered this, and this ash bed is point to the same period of time when we became bipedal. And these are a testimony to an upright gait. Again, I move on 2 million years. About 2 million years ago, not only did we become bipedal, we started to use tools. And these were stone tools that we used, and we started to move around because they were, after we evolved, then we moved around, and there are multiple fossils of Homo erectus that has been found in Africa and as far away as in Java. So we were quite mobile even about 2 million years ago. So this use of stone tools meant that we could see predators from a distance, and we could ward off predators or even kill the predators, which meant that we immediately gained the possibility of surviving longer, and the probability after birth that we survived became suddenly enhanced. And so this provided us with the possibility of Darwinian natural selection, and we became a more fit species, or not we, but Homo erectus became a more fit species, and we actually evolved from the Homo erectus later. Once the first species in this genus Homo, and we are Homo sapiens, but this is Homo erectus that I'm talking about, once that particular species arose, it began to spin off new varieties of Homo, such as Homo neanderthal, Homo heidelberg, etc. So they have been given names, and we, like I said, are Homo sapiens, and we are the newest member in this particular genus. And we have a large number of ancestors, ancestral species that belong to the same genus. There are now 21 known species of the human, and more are being discovered. The latest that has been discovered is called Homo longi, which has been discovered only about 3 years ago in 2018. So we believe that there are many species of Homo, and we probably have not discovered all of them yet. There are 21 known species. Like I said, that I'm jumping ahead, and I'm going to focus on this particular tree. This tree, as opposed to the previous tree that I have shown you, this tree is like a real tree. It's rooted at the bottom, so the base of the tree is at the bottom, and we climb up this tree, and we find that there are different branches of the tree. I'm going to focus on one particular set of branches of this tree. This branch that's marked as heidelbergensis. It's also known as Homo heidelbergensis, which is the name of the species of humans, of hominins that existed, and it's popularly known as the Heidelberg man. The Heidelberg man was ancestor to the Homo sapiens, to the Neanderthal man, and to something that you may not have heard called the Denisovans. And my story is going to spin around these three species, the Denisovans, the Neanderthals, and the Homo sapiens. And the reason why it's going to spin around that, although it does not cover all of human evolution, this is where Svante Pääbo, this year's Nobel winner, has actually contributed in a big way. And so I'm going to spin my story around these three. These three species. Homo heidelbergensis, our ancestor, as you can see from this reconstruction of fossil remains, look pretty much like us. And there was, if I didn't tell you the name, we probably would not be able to distinguish it from a modern human. So they look more or less like us, and from them arose three different species: the Homo sapiens, Neanderthals, and Denisovans. That particular branch that gave rise to the Neanderthals and Denisovans arose about 400,000 to 300,000 years ago. Now, whenever I mention these dates will have a large bracket, because most of the time the evidence is quite fragmentary in terms of a broken skull, a broken bone, etc. And therefore, we are never absolutely certain of the dates. It has a large bracket. So this is a bracket of 100,000 years. From the Homo sapiens, the modern humans, arose about 150,000 to 130,000 years ago. And so those are the two major branches that we're going to talk about. And like I said, that most of these kinds of evidence came from fragmentary evidence, broken skulls, broken bones, etc. And when we learned to analyze DNA, our evidence became more robust. Our evidence became more foolproof. So let me just quickly, for the sake of those who are not in this particular domain of science, let me quickly explain that each human DNA, first of all, we have cells, and inside the cell there is the chromosome, and inside the chromosome there is the human DNA. So packaged within each chromosome. The human DNA contains information for production of protein, various kinds of proteins that are required for our daily lives or our physiology, for warding off various kinds of pathogens, makes us resistant to pathogens, also determines our behavior, and so on and so forth. If I look at the DNA and if I sort of join the DNA end to end, it's a linear string that has about 3 billion alphabets. So the human DNA has about 3 billion alphabets. It's not one single linear string, it's packaged into different chromosomes, but for our purpose, it's good enough to think about a linear string, a linear sentence, a very long sentence containing 3 billion nucleotides. If we compare the DNA of any two individuals, we would find that we are largely similar, and we compare the two strings obtained from two different individuals, and if we compare them alphabet by alphabet, we will find that we are 99.9% similar. So we are only different. The difference between me and let's say another person who is in the audience, the difference in the DNA alphabet by alphabet will be about 0.1%. But 0.1% of 3 billion nucleotides means that we are different in about 3 million alphabets. And that difference of about 3 million alphabets gives us our individuality in terms of our behavior, in terms of how we look, in terms of our ability to resist diseases, and so on and so forth. So it's 0.1% is a large number of alphabet differences, is what I'm emphasizing. And how do these differences come about? Again, I'm going to cut a long story short. There are multiple ways that these various kinds of differences come about. We get exposed to different kinds of environments. Some of us smoke, some of us stay in high altitude areas, some of us expose ourselves to rays of the sun for long hours of the day, and so on and so forth. All these are DNA damaging agents. The DNA damage takes place almost constantly, but they are also repaired by various kinds of enzymes that are present in the body. However, sometimes these errors are not corrected, and if the errors are not corrected, since the DNA gets passed on from parents to offspring, whatever DNA accumulates in my sperm and eggs and so on and so forth, they will be passed on to the children, and therefore these errors get propagated, and over a period of time the errors accumulate, and in the course of human evolution, errors have accumulated to the extent that any two individuals are about 3 million alphabets different. So when these errors keep accumulating, a new species is born when the number of errors that have accumulated, number of changes are far too many for the two individuals within that group to mate and to produce viable offspring. That's when a new species is born, and etc. So I'm not going to get into the details of that. What one could do is, by studying the DNA sequences of various species, we can infer which species evolved from which species. In other words, what is my ancestral species I can infer by studying the DNA molecules. Also, since the changes accumulate over a period of time, and we sort of have a calibration of how quickly these changes accumulate over time, by noting the number of differences between any two DNA sequences, not only can we tell that which DNA was derived from which DNA molecule, but we can also estimate when this difference may have arisen or how many years may have elapsed since these differences have arisen. So the DNA evidence is quite foolproof. It's large data. It provides us with a handle on speciation. It provides us with a handle on ancestry. It provides us with a handle on time of evolution. So there are great advantages in using the DNA in order to understand not just human evolution, but evolution in general. And this is what Svante Pääbo did. He analyzed DNA of various species in order to draw inferences about human evolution. In particular, about settling a debate that reached the human evolutionists for a large number of decades, as a matter of fact. So let me quickly point out where we evolved, and again, much of this comes from DNA evidence. So we evolved in Africa, and how do we know we evolved in Africa? So let me provide a logical basis of this. Like I said, that mutations or these errors or differences accumulate over a period of time. So if a population group has evolved for a long period of time, between individuals in that population group, you would find larger number of differences. This is the expectation that we have, that older populations should possess a larger number of differences between individuals within the population among the individuals, the population that's evolved more recently, we would find a lesser amount of genetic diversity. So this is the expectation. In science, we often turn the logic the other way around. If we see if this is our expectation, and the expectation and the observation agrees with the expectation, we can turn the other side of the coin and say that those populations that have a great genetic diversity are the older populations. And this is exactly what has been done. If we look at, we estimate genetic diversity by sampling, by recruiting individuals in our study, sampling the blood, isolating DNA, and then sequencing the DNA in order to understand how much of diversity is there between individuals within that group. If we look at the total amount of diversity, what we find is that in Africa, the populations of Africa, or the individuals who live in Africa today, have much greater amount of genetic diversity than populations in Asia or individuals living in Asia. And those two are, of course, those two genetic diversity in those two regions are, as you can see, much higher than in Europe or Australia or New Guinea. This has been done in multiple ways, and so essentially the inference from here is that African populations are older, Asian populations are second most old, and the other populations are more recent. Populations of Europe, Australia, and so on are more recent populations. We cannot find any geographical region where there is greater amount of genetic diversity than Africa. So the most logical conclusion is that we must have evolved in Africa, and this logical conclusion is also bolstered by various kinds of information or evidence that we have gathered from our ape-like ancestral species, which I will not get into. Again, I'll skip this. It makes the same story. So there are, if you look at signatures of DNA, signatures in the DNA, there are many signatures that one can find. Some of these signatures which only the males have, and they pass on to all their sons. The father passes on to all their sons, and these signatures are a constellation of some alphabets in the DNA, in the DNA that's contained in the Y chromosome, and these signatures also have ancestors and descendants, and these signatures can be actually timed and dated. I won't get into the details. Suffice it to say that those signatures in this particular tree that you see, the ones that are A, B, C, D on the left, those are the older signatures compared to R1A, R1B, R, those are the newer signatures. If you plot these signatures, or the frequencies of signatures in populations residing in different geographical regions, this is what one sees. As you can see that, these light yellows, etc. These older signatures are all embedded in Africa. Populations in Africa carry those old signatures. If you come to the New World, the Americas, what you find is, for example, predominantly the signature Q, which is a more recent signature. So the oldest signatures are in Africa. The newest signatures are in the New World, which is the Americas, and the middle-aged signatures are all in Asia and Europe. So this is consistent with the theory that humankind evolved in Africa. They came out of Africa, walked, traversed in Europe, and crossed the Bering Strait and went and peopled the New World. So this is consistent with this hypothesis to begin with, but this is very strong evidence to infer that humankind evolved in Africa, moved out of Africa to populate other parts of the world. This evidence comes from paternally transmitted signatures. If we look at maternally transmitted signatures, again, the names of the signatures are different, like L1, L2, L3, and so on. But the ones on the left-hand side are the older signatures, the ones on the right-hand side are the newer signatures. And if you plot these signatures by sampling individuals from various geographical regions, you essentially see the same story. The oldest signatures are in Africa, and the newest signatures are in the New World. The middle-aged signatures are in Asia. So the maternally transmitted signatures are in what's called the mitochondrial genome. I will not get into the details of it, but essentially it's transmitted by the mother to her children. And if you look at that, those signatures, again, it's consistent with the theory that humankind evolved in Africa, moved out of Africa, and traversed Asia and Europe and went and peopled the New World. So this is by and large what we understand about human evolution. If you look at origin of modern humans, when did we evolve in Africa is the question. So there's no dispute over the fact that we evolved in Africa. Various pieces of evidence. I'm providing you with genetic evidence because that's what my bread and butter is. People who work on fossils and bones and so on and so forth, they will provide you with other kinds of evidence, and all of this sort of synchronize. All of the inferences from various domains of knowledge actually synchronize, and what we find is that modern humans, anatomically modern humans, evolved in Africa, and the time of evolution of modern humans is about 150,000 years ago. It could be 130,000 years, it could be 140,000 years, but like I said, that these dates will have a large bracket. So anyway, this particular skull has been again discovered by a member of the Leakey family that I talked to you about. This particular skull was identified or discovered in Ethiopia, and again, radiocarbon dating, etc., dated this particular skull to be 130,000 years. And this skull certainly anatomists and people who study human anatomy, etc., will vouch that this belongs to a modern human. So the best estimates that we can get of modern humans having evolved in Africa is about 150,000 years. Like I said, that we evolved in Africa, moved out of Africa to populate other parts of the world. One of the places that we populated almost immediately after coming out of Africa, within about, let's say, 50,000 years or so, we came and peopled India. So if you look at Indian populations and if you look at diversity of Indian populations, after Africa, the Indian subcontinent has the highest amount of genetic diversity of its individuals, of its populations. And so India India has a center stage, occupies a center stage in human evolution almost after Africa. So what we did was like I said, that we came out of Africa and started to walk around and explore other places and move over geographical regions, ultimately moving into the New World. But when we came out of Africa, we found we were expecting that we were the most evolved species and there were no other species staying outside of Africa, but that's not what we saw. What we saw was that we came out and saw that we were not alone. There were other kinds of species that were living outside of Africa in Europe and Asia, and those species also looked very much like us. Who may have been those species? Well, we know that the Neanderthal man lived in Europe for sure. So we met the Neanderthal man. And then later, at some point of time, these people who were hunting fossils and looking at trying to find bones from which to extract DNA, they found a cave in Siberia, and they isolated the DNA from there and discovered that whoever was living in that cave, or the family that was living in that cave, they were not Neanderthals, they belonged to a different species. And that cave is called Denisova, and so they named the new species as Denisovans. So that's all fine, but so what is the controversy? And the controversy that raged, so like I said, that we met at least two other species of hominid cousins. One is the Neanderthal, and the other is the Denisova. The Denisova is a cave in Siberia. They found a finger bone. They were able to isolate the DNA and sequence the DNA and show that that particular sequence was too far away from the Neanderthal genome, and therefore that must be a separate species. Like I said, by counting the number of differences between two DNA sequences, you can guess, you can figure out whether the two DNA sequences belong to the same, belong to two different individuals of the same species or belong to two different species. In this case, they were able to figure out that they belong to two different species, and the new species from the finger bone, the DNA that they isolated, they said that it belongs to a new species, and they just called the species the Denisovans, because that cave named as the Denisova cave. So I said that the Neanderthals were recognized as a separate ancient human group. More than 100 years ago, they were recognized as a separate ancient human group, as a separate species. They almost lived for 1 million years in Europe and Western Africa, and suddenly, about 30 to 40,000 years before the present time, there was extinction of the Neanderthals. They disappeared from Africa, from Europe, from the face of this planet. So one of the major questions that that people thought, and that is the debate that raged for a long period of time, we modern humans were better tool users, even though Neanderthals were also tool users, but we were probably better tool users. So it's quite possible that we used tools of various kinds to actually kill Neanderthals, and we exterminated them. We must remember that one of the reasons why we started moving out is because our fertility was better. We were tool user. We could use stone tools, and therefore our probability of survival was higher, and therefore ultimately in a particular region we reproduced, and we over reproduced, and there was pressure on natural resources. We were still hunter-gatherers, so we were essentially exploiting what was available in the nature, naturally around us, and so there was pressure on natural resources. There were too many mouths to feed for the amount of food that was available in nature around there, wherever we were living, and therefore, in order to feed our children, we had to move out and explore new territories, explore new natural habitats. So that's when we came out of Africa in exploration of new habitats. So we were larger in number, we were better tool users than the Neanderthals, and we probably clubbed them to death and we exterminated them. So that was a popular theory. And we also knew, again, by various kinds of anatomical, fossil evidence, DNA comparisons, that DNA and the Neanderthals were a direct ancestor of the present-day Europeans. So they provided, the base biology of the current-day Europeans, but the Europeans probably, beat them to death and exterminated them. So that was the popular theory that remained. However, Svante Pääbo actually killed that particular theory through his various kinds of findings. He actually was not a believer of that theory. He didn't believe that modern humans were so violent that they could kill thousands of Neanderthals. And so he didn't believe that. So he set out to test his own hypothesis, which I will come to in a minute. And eventually was able to prove that he is right and we didn't kill the Neanderthals. And that's the reason why he got the Nobel Prize, not just for disproving a popular hypothesis that raged for a long period of time, but also in order to disprove that hypothesis, the kind of technologies and concepts that he had to provide to the world of science was something that's awesome. And it's because of those awesome feats that he actually, in conjunction with his proof that we didn't kill the Neanderthals, together and concomitantly with other kinds of evidence that he provided on human evolution, he was awarded the Nobel Prize for Physiology and Medicine. So this is a cave in Croatia. Croatia is near the Adriatic Sea. And this particular cave in Croatia, they found a fossil, And from that fossil, they found 3 bones. And these bones were again radiocarbon dated to be about 40,000 years old. Remember, the Neanderthals became extinct sometime between 30 and 40,000 years ago. So these are the 3 bones, and they belong to the Neanderthals about 40,000 years ago. And the Svante was able to isolate DNA from these bones, 40,000-year-old bones, and isolating DNA molecules was by itself a big chore. But he was able to do it, and the DNA was very broken, the DNA was very fragmented, so he was not able to sequence the entire genome of the Neanderthal, but was able to sequence it in bits and pieces, sufficient enough to be able to draw useful conclusions. So again, these are the 3 technical slides, and I'm not going to get into the technical slides, just to mention a few things. Number 1, the DNA is very fragmented when the DNA is old. When we isolate DNA from a bone or from a skull, etc., these are very fragmented, and only short fragments can be sequenced. So that's one major problem. The other problem is that because of vagaries of weather, etc., there are some alphabets that get artificially changed. These are chemical changes that happen, and these are not what the whoever that DNA came from. That person did not have those specific alphabets. The alphabet got changed after the person died, and essentially due to various kinds of chemical modifications. And that those chemical modifications, if you didn't take care of those chemical modifications, you would unnecessarily ascribe certain wrong alphabets to the person from whom this DNA was taken. And therefore, these kinds of corrections had to be done. First of all, the recognition that these kinds of chemical changes can happen in the DNA even after a person is dead. And to be able to recreate, to be able to correct for those changes needed a huge amount of work and a huge amount of intellectual inputs. The other is that these are various kinds of changes and various kinds of changes that happens. The other is for those of us who do this kind of analysis, and I know that there are many people who are conversant with this. We can't actually sequence a piece of DNA, without actually making multiple copies of itself, a process that's called amplification. And this amplification is hindered when you isolate DNA from a bone, and you need to then overcome these various kinds of hindrances, the challenges that you face, and these challenges were overcome. These challenges were faced by Svante Pääbo, and he actually overcame those challenges to be able to analyze ancient DNA. I will also skip this essentially. I'll skip this. So ultimately, Svante Pääbo was able to isolate DNA from these 30, 40,000-year-old bones, and was able to make appropriate, face the challenges, overcome the challenges of amplification. And then when you sequence, then you also need to make corrections for the fact that many of the alphabets that you're actually seeing in your DNA sequence are actually wrong. They are because of chemical modifications. They are not original, so you have to identify those. You have to discount those, etc., and to be and ultimately get hold of the sequence of an individual who lived about 40,000 years ago. Also, you needed to identify which particular alphabets belonged to the ancient individual, and which alphabets belong to us. So in this set of humans, these are all human DNA sequences, and this sequence of human DNA sequences, those blue alphabets belong to us. They are indigenous to us. They belong to us. The red ones don't belong to us. And again, there are various ways of telling why the red ones don't belong to us. Those red ones came from some other individuals, and those individuals were Neanderthals. So those red ones are Neanderthal specific DNA alphabets, and the blue ones belong to us. So those are indigenous to us, the red ones are external to us, and those external alphabets were provided by the Neanderthals to the humans. How can the Neanderthals provide nucleotides to the humans? The only way that Neanderthals could have provided these nucleotides to the humans is if they had mated with the humans. So that's the only way that you can exchange these kinds of nucleotides between two DNA molecules belonging to two different species. So from here, as soon as those specific nucleotides belonging to the Neanderthals were identified and they were found in the humans, the immediate conclusion was that Neanderthals and humans mated with each other. I will tell you more about what happened to their children, but from this, the foolproof evidence was one of mating, one of reproduction between Neanderthals and humans. So that was something, again, very interesting, that Neanderthals and humans mated. One had not conceived of this. The fossil hunters, the paleontologists, were not able to come to this conclusion. It was only possible by analyzing DNA, and this was a very painstaking effort of over 10 years that Semantic actually worked on. So that was, having said this, that you have Neanderthals and humans who are reproducing, who are producing children. How could the humans actually club the Neanderthals to death and exterminate them? That was inconceivable. So as soon as Svante was able to show that we have Neanderthal genomes in our own genomes, and the only way that it could have come is through mating, is through reproduction, immediately the conclusion was that it's very unlikely that we actually beat them to death, beat the Neanderthals to death. What happened then? How did they become extinct? That's a story that will unfold soon. So ultimately, from this draft segment sequence of the Neanderthal genome, it was quite clear that the Neanderthals were closer to non-Africans than to Africans. They were closer to non-Africans. I told you that Europeans descended from the Neanderthals, so they are Europeans and non-Africans. The Neanderthals were closer to non-Africans. Another revealing feature that came out of this draft sequence that was published in 2010 by Fable's group, the direction of gene flow. I said that the Neanderthals and the humans mated with each other, so the Neanderthal portion of the Neanderthal genome actually came to the humans. But if they mated with each other, then we would expect that some of the human-specific nucleotides or human-specific portions of the DNA would also go to the Neanderthal, but that did not happen. The direction of gene flow was unilateral from Neanderthals into modern humans, but not the vice versa. Again, people scratch their heads, and the most parsimonious explanation of this unilateral gene flow or unidirectional gene flow is that the Neanderthals and the modern humans mated. They procreated children, and the children then did not go to the Neanderthals, they went with the band of humans. And so the children who had a mixture of Neanderthal genome and the human genome, because they went to the humans and did not go back to the Neanderthals, so we couldn't find Neanderthals having modern human genomes. So, but the modern humans had Neanderthal genomes because the children came with the modern humans. So that's the best case scenario, best case explanation that we have as of this point of time, is that humans and Neanderthals, modern humans and Neanderthals procreated, and the children went with the humans. This immediately, if this is to be accepted, this immediately also led to a clean hypothesis how the Neanderthals could have vanished. Imagine that the children they are making, imagine that the children are going with the humans, the human group is expanding, and the Neanderthal group over a period of time would obviously decrease in numbers because they would die. The children didn't go back to them. The older Neanderthals would die. The children did not go back to the Neanderthal group. So over a period of time, through deaths and through other kinds of natural calamities, the Neanderthals would shrink in size, and over a period of time, they would actually be exterminated. So the point that Semantic made, or the hypothesis that that he proved or disproved, the hypothesis was that we clubbed them and beat the Neanderthals to death. He disproved that, and he said that that's not how it happened. We actually embraced them, and we actually absorbed them into our group, absorbed the children into our group, and that's how they became extinct, and we came to stay and flourish. The extent of Neanderthal ancestry is between 1 and 4%, meaning that the extent of Neanderthal genomes in our own genomes is between 1 and 4%. They eventually was able to identify, isolate DNA from bones. from the Altai Mountains, and they were able to obtain the complete genome sequence of the Neanderthal. And this was 4 years later. That was 2010 when the draft sequence was published, and this is 2014 when the complete genome sequence of the Neanderthal was published. Very interesting, in science, when you meet these roadblocks, you will get very, very frustrated and make certain statements. You think that these are prophetic statements. Later, you have to eat back your own statements. So these are two statements from Svante Pääbo, who was frustrated because he was unable to amplify the first extract, ancient DNA from the bones and then amplify them and sequence them, etc. So this is what he said, and he wrote, as a matter of fact, we'll never be able to sequence the nuclear genome of a Neanderthal. It's too degraded. The vagaries of the weather, etc., will make the DNA very fragmented, and it will degrade the DNA, and therefore he said that we'll never be able to sequence the nuclear genome. And then he said that there's too little DNA there, it can't be done, it can't sequence them. Later, he was to eat back his own words. And of course, this is the Cold Spring Harbor laboratory lecture that he was giving, a named lecture that he was giving called Genetic Legacy of the Neanderthals. This is in 2010, 5 years after the draft sequence and the year after he obtained the complete sequence of the Neanderthal genome. And he said that, of course, you should never say things like that, particularly in biology, because you'll often be overtaken by technical developments. And in this particular domain of knowledge, or generating this kind of evidence, he was the architect of these technical developments. And they were not easy technical developments. They were very hard technical developments. So you will often be overtaken. He's the one who actually overtook the rest of the world through these various kinds of technical developments that made possible the sequencing of the Neanderthal genome, even though frustration shows from for about 10 years that he was trying to sequence the Neanderthal genome. Many Neanderthal genomes have since been sequenced. The total number exceeds 8,000. So this is August 2022. Almost 8,000 Neanderthals have been sequenced. And as you can see that they have been sequenced primarily from the colder region. If you look at India, you don't see it's completely blank. And so the Europe and other parts of Europe, most parts of Europe were like deep freezers. So the Neanderthals died, and immediately they got buried in snow. So they were placed in deep freezers, and therefore their remains were not so quickly degraded. Unlike in India, when they may have died, if at all they roamed about the streets of India. The Neanderthals, that is, they would have died, and because of the climatic conditions here, the humidity, the rain, and so on and so forth, the bacteria and fungi would have grown on them, and obviously those bones and skulls would not have been preserved very well. So it's most of the DNAs that of the ancient Neanderthals that come from are from various places in Europe. I showed you this particular cave called the Denisovan cave, which is in Siberia, from where a finger bone was isolated, and again, from the finger bone, DNA was isolated, and that DNA led to a complete genome sequence of this particular individual that's been called the Denisovans. So again, Svante Pääbo and another person whose name I must state, his name is David Rees. David and Svante were able to together join the two labs and sequence the Denisovan genome. The Denisovans had an evolutionary history which is distinct from the Neanderthals and modern humans. I won't get into the evolutionary history. Suffice it to say that the Denisovans were a separate species from that of the Neanderthals. Near East about 50 to 6,000 years ago, and then we probably lived somewhere in the Middle East before continuing on to Eurasia, to other parts of Eurasia. There were also some amount of admixture with the Denisovans in Southeast Asia and elsewhere. So one of the and then the Denisovans and the Neanderthals became extinct between 30 and 40,000 years ago. but they continue to live. As a matter of fact, Neanderthals, both Neanderthals and Denisovans, even though we don't get their physical structure, they continue to live, and their genomes continue to live in us. So we have about 1 to 2% of modern humans of now African ancestry, and up to 7% of the genomes of people in Melanesia. So Neanderthal and Denisovan genomes actually have between 1 and 7% of people in different regions of the world, and the Neanderthals and Denisovans actually live on through us because their children went with us. The children of meetings between Neanderthals and humans, or Denisovans and humans, went with the humans and did not go back to the Neanderthal group or the Denisovan group. So they live in us. They continue to live in us. I'm not going to get into this details of this particular study that's been done very, very recently, about 3 years ago. I want to focus on one particular sentence, which is that these guys were able to find bits and pieces of DNA in the humans, which did not belong to the humans, which did not belong to the Denisovans or to the Neanderthals. So they were pieces of DNA that came from some kind of an archaic hominin that's never been identified yet. So they were derived from as yet an unknown archaic hominin. This will bring us a little bit to our own studies on human evolution in the Indian subcontinent, and I want to share a little bit of our results with you. So we were working on various population groups of India, trying to reconstruct population structure, human evolution in India, and we went to the Andaman and Nicobar Islands, where, as you know, some African living tribals who look like Africans live in the Andaman and Nicobar Islands, and those tribal populations are called Jarwas and Ongis. They are very small in size. Their demographic size, numerical size is small, but we were able to obtain blood samples from some Jarwas and some Ongis, and we were able to isolate DNA. They were living Jarwas and Ongis. There was no problem of isolating DNA. We sequenced them. We analyzed the DNA. And again, I will not try to explain this entire picture. These yellow pieces that we find among the Jarwas and Ongis, which do not belong to the humans, did not belong to the Neanderthals or to the Denisovans, but were timed around the same time. The DNA molecules evolved around the same time that Denisovans and Neanderthals lived, but they did not come from Neanderthals or Denisovans. So we also postulated that it must have come from an archaic hominin which has not been identified yet. And we published this in a reasonably famous journal called Nature Genetics. Won't get into the details of this, so this is our analysis, comparative analysis of the DNA sequences of Jarwas and Ongis with that of Neanderthals, Europeans, Denisovans, and major peoples of Southeast Asia, major population groups of Southeast Asia. Simultaneously, so we published this paper in 2016 September. In October, there was another meeting, another major meeting of the American Society of Human Genetics, where people who were studying human evolution in Melanesia, which is this group of islands off of Australia, they conducted study among these tribal populations of Melanesia, and they also concluded that genetic study reveals ancient Melanesians integrated the mysterious hominid. So as we can see that various studies, genetic studies, are finding bits and pieces of DNA in our own, in modern humans, that do not belong to us, not to the Denisovans, not to the Neanderthals, and as a matter of fact, do not belong to any of the known species of humans. They belong to mysterious hominins. So again, bolsters the view that humans, there were multiple species of humans. Modern humans or in our ancestral species were very many, and we haven't actually identified all of our ancestral species. One of the questions then that we asked is that, okay, so we find that 7% of the DNA in Melanesia belong to of modern humans belong to the Neanderthals or the Denisovans. How much do Indians have, how much of Neanderthal and Denisovan DNA do modern humans living in India have? We conducted this study in Asia. I'm not going to give you the entire data set, primarily because I want to focus on India. And we looked at about 42 ethnic populations of India. We sampled about 600 individuals. We did whole genome sequencing, and we were able to estimate the proportion of Denisovan and Neanderthal genomes in our own genomes, our meaning those of us who live today. And what we find is that our genomes have Neanderthal DNA to a certain extent, and that varies from one population to another. On the x-axis are names of population groups or abbreviations of names of population groups that I will not expand right now, just for the sake of time. And on the y-axis is the percent of Neanderthal genomes in our own genomes, and that varies between 2 and 2.5%. So on an average, we have about 2% of our DNA that actually does not belong to us, belongs to the Neanderthals. So the Neanderthals live on through us. Denisovan admixture, less, about 0.08 to 0.4%. So it's about 0.2%, let's say, of the Denisovan. So 2% of Neanderthal admixture, 0.2% of Denisovan admixture in the modern human genomes of people who live in India. The last question that we want to address is that, okay, the Neanderthals and humans mated and procreated. The Denisovans and humans mated and procreated. Did the Denisovans and Neanderthals mate and procreate? The answer is yes. It was only a matter of time. We knew that that was coming. It was only a matter of time. Again, Subhatepevo was in some ways lucky, in some ways had an observant eye, and people were collaborating with them. So I'm not going to again tell you the entire story, except to say that this Denisova 11, which is half blue and half red, the blue comes from the Neanderthals, the red comes from the Denisovans. As we know that when we are born, we get half of our DNA from the mother, half of our DNA from the father, and so we are a 50-50 juxtaposition of the genomes of our parents. And so that's exactly what we find in Denisova number 11, which was isolated from a bone in the Denisova cave, and that's a testimony to a child being born of a Neanderthal, whatever, one parent Neanderthal, the other being a Denisovan. They went a step forward because they were able to isolate DNA from X chromosome, or identify DNA from the X chromosome and Y chromosome, and they were able to infer that the Neanderthal, this particular individual had a Neanderthal mother and a Denisovan father. Just saying 50-50 does not identify which parent is what, but they were able to identify the parents as well by analyzing the DNA in a deep way. I will come to the last 1 minute of my talk, which is that I said that this person, Subhatepevo, actually got a Nobel Prize in Physiology or Medicine. And although his citation says that he got it for providing an understanding of human evolution, but he also, this understanding of human evolution, which portions of our own genomes come from the Neanderthals, or which portions come from the Denisovans, etc. They also bear some stories on our susceptibility and resistance to various diseases. He provided multiple examples, he meaning Subhatepevo, provided multiple examples, but I just want to spend 1 minute on this particular risk factor, which predisposes us to getting severe COVID-19. And this, many of us have been exposed to SARS-CoV-2. Some of us were asymptomatic, some came down with very minor symptoms, but some had to be hospitalized, and many actually were hospitalized and died because they had severe COVID-19. Ultimately, it was identified that there are pieces, there are portions in the genomes that actually make us susceptible to severe COVID-19 once we are infected with SARS-CoV-2. Not all of us possess those susceptibility factors, but what Subhatepevo and his group was able to do is that they identified these susceptibility factors in our genomes that make us susceptible to severe COVID-19 when we are infected with SARS-CoV-2 actually comes from the Neanderthals. So the Neanderthals provided us with that particular segment of the DNA that predisposes us to severe COVID-19 if we are infected with SARS-CoV-2. Again, I'll skip this. If we look at this is essentially the final slide. If we look at the frequency of this particular segment of the DNA that predisposes us to severe COVID-19 on infection with SARS-CoV-2, what we find is that in India, the proportion, the red portion is the predisposing segment of the DNA that predisposes us to severe COVID-19. And the frequency of that particular segment is very high, as you can see from this graph, is very high among people in India. So we are particularly predisposed to getting severe COVID-19 once we get infected with SARS-CoV-2, which also explains why we had a huge number of deaths in India. Unfortunately, this is the segment that we actually inherited from the Neanderthals. Okay, so this is what my story was, and I will justify my title. We moved, we embraced, and we absorbed. We moved out of Africa, and I hope that I've been able to justify that. Once we moved out of Africa, we met Neanderthals, Denisovans, and other species of hominids. We embraced them so tight that we actually interbred with them, and we absorbed them. The children of these meetings actually went with us and did not go back to the Neanderthals and Denisovans. So that justifies my title, and we moved, we embraced, and we absorbed. Thank you very much for your attention.
Hello. Hello.
Yes, I'm able to hear you, probably.
Yes. Mesmerizing talk. Wonderful.
Thank you. I hope that you could hear me. I was afraid that you were unable to hear me, but I'm glad that you are able to hear me.
Yes, I could. Well, Arijit, are you there?
Can you tell him?
He's muted, but I do see him.
Well, I cannot express my gratitude and thanks to you for this wonderful, mesmerizing talk about our evolution and ancestry. I am not an expert in the field, but I could understand the whole story. Once again, I thank you, Professor Majumdar, on behalf of our trust, our audience, our visitors, and our attendees, that they must be benefited by your wonderful talk.
Yeah.
Arijit, you want to say something? Arijit, are you there?
I think he just logged out.
Can I don't know whether Professor Majumdar will be able to take any question answer.
I'm happy to take questions, no problem. I'm happy to take questions.
Okay. Any question for Professor Majumdar? He will be happy to answer from the attendees.
I'd like to ask
May I ask a question?
Yes,
I'd like
please
to
go ahead.
ask you.
Please, one by one.
Jyoti, please go ahead. Jyoti, go ahead.
If I understood correctly, I mean the Asians probably interbred with another hominid species. Am I correct?
That's absolutely right. Yes.
Yes. So, is there any conjecture where these hominid species may have originated from, and do we have any clue where the evidence may be found from fossils or whatever of this different hominid species who might have interbred with the Asians?
No, Professor Bose, it's very difficult to speculate, primarily because many of these fossil remains, especially in regions where it is hot and humid, they did not survive very well. So, like I said, that we have already identified 21 different species of humans. They came from mostly colder regions. So, if we mated with an unknown hominid, it's quite possible that that hominid lived in India, and their fossils probably won't be preserved so well. So it's very, very difficult to speculate which geographical region we might be able to find this unknown hominid with whom we mated and procreate.
Okay, thank you.
Any more questions?
Yeah. I'd like to ask one thing. if we follow the Darwin's laws of evolution and Lamarckism, is there any effect was observed among this observation that you presented with the evolutionary strategies of the Neanderthal, Australopithecus and Neanderthal in Australia, that they were also being affected by the environment, and in the evolution that in the fossil studies they were recognized on the basis of the environment as well as the evolution, any aspects of development. If you could understand, you say yes, or if you not understand my question, I can once again repeat, any other influence of Darwin and Lamarck can be studied on your study.
I don't know about Lamarck, but certainly Darwinian evolution signatures or footprints have been found in genomes. So we might ask this question that, the way the natural selection works, for example, one particular way in which natural selection works is to protect us when we get exposed to pathogens. So many of us are able to survive exposure to various kinds of pathogens. Some of us succumb and die. So there are portions of the genome that actually are under intense effects of Darwinian natural selection, either positive or negative, and not the entire genome is not subjected to Darwinian natural selection. So the question is that, can we identify which regions of the human genome have been subjected to stronger effects of Darwinian selection? And people have actually asked that question, and we have been able to identify regions of the human genome that are under intense pressure of natural selection, positive or negative.
The Lamarck is not coming in this picture.
Well, Lamarck, I don't know. Lamarck talked about use and speech of characters. Lamarck is actually coming in through the epigenome.
Okay.
Which actually, again, those views are still crystallizing, and the Lamarckian views and are actually still not entertained very well by biologists. But there are some thoughts about Lamarckian possibilities through the epigenomes of different kinds of species. But we still don't have very strong evidence of Lamarckian pressures.
So now the present world with the corona and the COVID, because they're mostly burned and they have already changed in ashes, any other evidence will reflect our future, that there was a dangerous time that we have crossed in the evolution, that's 2019 to 2022, regarding the millions of fatal expired people were in this world.
No, I'm unable to hear the question. Your voice is cracking and I'm unable to understand you.
There will be any existing evidence of so-called in the evolution, take it thousands years back or future, what would be actual our document? Because the most of the fossils or most of the bones have been burned, and there will be no trace of this COVID.
Trace of COVID, you mean?
Yes, yes.
Again, you are right that people who die or who have died, they have been buried or burned, whatever. In Europe, they have mostly been buried, and I don't really know whether many years later, many thousands of years later, you can actually isolate SARS-CoV-2 DNA from those bones of those individuals. I doubt it. Because they usually will be in the lungs of these people, and lung is a soft tissue. Most of the time, the lung will be gone. So I don't think 1,000 years. Looking forward 1,000 years, we will be able to identify SARS-CoV-2 genomes in individuals who actually died from severe COVID-2. I really honestly don't think so.
Yeah, I mentioned genetic sequence could represent that many years evolution in future, that we have crossed a tremendous time of 2018 to 2022 or 21. The genetic events will remain. Am I correct?
But even those genetic events may be very difficult because at least SARS-CoV-2 genomes won't be identifiable because they will mostly be present in the lung tissue, and lung tissue is soft, and therefore it will be gone. It doesn't hide in the bones. SARS-CoV-2 genomes, they don't infect the bones; they infect soft tissue. So it's unlikely that we will be able to isolate SARS-CoV-2 genomes 1,000 years later from an individual who died of severe COVID-19.
Thank you very much.
Thank you for the question.
Thank you, sir.
Any more question? Well, Arijit, are you there?
Yes.
Arijit, now
Yes, I'm here.
please, it is your responsibility
Yes,
to
yes.
for thanks to Professor Majumdar for his excellent mesmerizing, illuminating,
Yes,
enriching talk.
we must. This has been a very thought-provoking, educative, informative, as well as illuminating discussion. And the person on whom we have discussed is Svante Pabhu. He has been awarded the Nobel Prize this year for his works. I had, in fact, one question. I was very curious to Professor Majumdar, that how long does such a research as Professor Svante Pabhu, which is done, take? Is it a lifetime research or a continuing research which like goes on through generations? What would be the kind of end, and when does such a research start?
Well, there is continuity in science, and so there's no start and end of this kind of research. But if we want to look at, Svante Pabhu's contributions to science, they probably started around the turn of the century, around 2000, is when he got this bright idea that he should try and sequence ancient genomes. The very thought of being able to analyze DNA from ancient genomes struck the minds of many people, but we essentially thought that it would be useless to do that kind of research because we are not going to succeed. But this man was brave. He stuck on to his ideas, even though he felt frustrated from time to time. He stuck on to those ideas and was able to succeed in about 10 years' time. So I would say his most productive period was 10 years when he got this thought and was able to actually get this thought into fruition by producing the first draft sequence of the Neanderthal genome. So, but generally, science has continuity and it goes on for generations, and we add more and more evidence to all of this, and that will continue. Analysis of ancient genome is continuing and will continue.
Thank you, Professor Majumdar. I thank all the attendees who have been present today, and my fellow trustees. Special thanks to Professor Parul Chakraborty, Navanita Law, and Vikas Chakraborty, who have organized this wonderful educative session. We thank all the other organizers who have made it possible today evening. Have a wonderful evening, everyone, going forward. And I'm sure we will all be thinking about how we are going to evolve in the coming days after this discussion, and what is there in store for the future.
Thank you. Thank you.
Thank you very much.
So I am bringing this session to a close.
Yes.
Thank you.
Thank you.
For participation.